Hello, everyone. Good afternoon, good morning to everyone. My name is Carmen Guguda, and I'm your host from Technovis. Here today, I have with me Marco van Rohr, a scientific specialist at CARBOGEN AMCIS. He is our presenter today for today's webinar, From Poly to Isothermal Solubility Data, Efficient and Fast Anti-Solvent Mediated Crystallization Design. Before we start, and I give the floor to Marco, I would like to let you all know that this webinar is being recorded and it will be made available most probably somewhere tomorrow. You will receive an email where you could watch, rewatch, or for those that they are not here today, they could watch once again the webinar. The same, I think Marco has agreed to also share his slides, so you will be able to download them later tomorrow from our publications database. The webinar will last about 30 minutes, up to 45, including also some questions. We will take questions at the end. But if you would like to later on still have a question and you would like to contact us, please feel free to write to us at info@crystallizationsystems.com, and we will try to address all your questions or to share your questions with Marco directly. I think Marco will also share his contact details soon. Before we go to the presentation, just for those of you that do not know that well Technovis, we are a Dutch company located in the north of the Netherlands, a world-leading technology provider for solid-state research, process development, formulation, and reaction screening. Since last year, we have 2 product portfolios. Most of you that are joining today are very familiar with our crystallization portfolio, but since last year, we have a second portfolio related to reaction screening. Our React-All instrument, a multiple reactor system with automated sampling, quenching, and diluting directly into the HPLC vial. It's your instrument for root scouting, reaction screening, optimization, reaction profiling, kinetics, you name it. I know that some of you already know the crystal breeder. It's our instrument from the crystallization portfolio meant to be used for growing single crystals. Crystal 16, our instrument for solubility, metastable zone determination, nucleation, induction time measurements, phase diagrams, and if you'd like to perform polymorph, salt formation type of screens, co-crystallization, even amorphous solid dispersions are possible with this instrument. I think you will also hear a bit more about solubility from Marco today. Crystalline is our slightly larger reactor of about 2.5 to 5 milliliter working volume. Is our instrument that comes with camera that works exactly like the camera technology that works exactly like a microscope, and also Raman. If you have a Raman spectrometer in your lab, you can easily connect it to the crystalline and follow crystallization formulation processes or chemical transformations with the Raman. And with the use of the images produced by the cameras, you can look at for habit or any type of experiments related to habit control, particle size, so process optimization, and so on. Comes also with an AI type of software that really gives you the benefit when looking at particle size and control. And this is everything about our instruments. And now I'm coming to our presenter today, Marco. As I mentioned today, he is a scientific specialist at CARBOGEN AMCIS. He started his career back in 2009 as a lab technician, taking care of synthesis of small molecules and peptides. Some years later, he studied industrial chemistry at the University of Applied Sciences in Zurich and got his bachelor degree in physical chemistry. CARBOGEN AMCIS, his first employer, and still after so many years, still working at CARBOGEN AMCIS. And as you could see, and you'll hear also from Marco, has a very vast experience and knowledge from early to later production pilot scale. He started in the production department where he learned a lot about hands-on chemical production up to 630-liter pilot scale. Really impressive. After this short-term production period, he moved to the R&D department where he was most of the time occupied with process familiarization and upscaling questions. 5 years ago, he moved from the daily business small molecule R&D to the CARBOGEN AMCIS specialist team. He started to intensively there work on crystallization development and profound characterization of solid phases. Most common challenges during his time in this team become evident during upscaling. So yeah, quite a vast experience at CARBOGEN AMCIS, many years with a lot of knowledge, a real chemical engineer in the field of solid state chemistry. Thank you very much for joining us today and bringing this webinar to our attendees. I think you can share now your slides.
All right. Thank you very much. Can you see my screen?
Yes, perfect.
Brilliant. So welcome everyone to my talk today about transformation of polythermal into isothermal solubility data and how to build crystallization processes out of that. I will initially start with a little presentation about a couple of topics about my employer, CARBOGEN AMCIS, where I work now in my 10th year, I guess, with. This will be the shorter part, and a little bit longer part will be about how to process solubility data and how to build quite quickly a process out of that. Good. CARBOGEN AMCIS is a globally operating company, so we have different size in Europe, so most of them are located in Switzerland, as you can see here. One of them in the UK, we have a site in France, one in Netherlands, and a couple of them in India and China. So what we do, I would describe CARBOGEN as some sort of a one-stop shop. We have sites focusing on early phase and raw material synthesis in Manchester. Very good to have this in the line, as they can provide us with starting materials out of the GMP sequence with a capacity out of 4.5 cubic vessels. We have the Swiss sites, they're more focusing on ZGMP manufacturing, and we can also handle high potent substances. I will say a word about that later on. And we have a capacity up to 2.5 cubic vessels here in Switzerland. We do some GMP flow chemistry, and I'm working in the department for API development and production. If you want to go on a larger scale, we have a site in Shanghai or also in India with large vessel capacity, also capable of doing cGMP synthesis. And at the very end, there is our Saint-Beauzire site in France, which is focusing on drug product manufacturing, also capable of doing high potent substances with a focus on formulation development, lyophilization, and aseptic filling. I listed here a couple of our equipment just to give you an overview how it looks like at our company. So we have a lot of multi-purpose equipment, for example, 70 general purposes vessels from 6,000 to 8,000 liter scale for GMP manufacturing, cryogenic capabilities, as well as high temperature reactors. So we do a quite big range of different reactors. We have a variety of autoclaves and filter options, and also capabilities of dry and wet milling, as well as pilot scale lyophilization for all kind of chemistry we I said in the beginning, we do hypo chemistry. So what we call hypo is highly toxic compounds, typically from oncology or oncological use. The highest category we have is 4+, so this is with a very, very low OEL of 0.05 microgram per meter cube. Very high toxic material. We can handle that at a couple of sites. In India, we do category 4 and some see category 4+. So this is really a standalone sign for CARBOGEN and one of our core business. Handling these type of materials, and I added here some nice pictures to illustrate to you how people work in these special containments, or with the glove box in front of a reactor. Everything is closed. There are also glove boxes, just to give you an idea how this looks like. We also do solid state for sure. The group or department I'm working, and I would describe my work built as on the 3 pillars. So we have crystallization design, which is, for example, how to developing a process which is about impurity rejection or yield optimization. We have a lot of screening work ongoing. Most of them is polymorphism and salt screens, but also solubility and stability screening, and sometimes we screen also for morphologies. One major part, which is also very important for me, is upscaling, and we have good capabilities to simulate what is going on plant. As Carmen said in the beginning, I've been working in the plant a couple of years, and I saw what happens if processes, especially filtrations, are not well designed, and it really teaches you about how time is lost and what we can do against it in production. As you all know, crystallizations are highly scaling dependent unit operations. For this work, we have a couple of special equipment. This list is not finished, but just to give you an idea, so for sure we have the Crystal 16 for all the solubility profiles, and then, for example, also for high frequent thermal cycling, as we use it for salt screens or form conversion screening. We also do continued crystallization, ultrasound assisted in a multi-stage crystallizer, so-called CSTR technology, which allows us to access kinetic product. We use PAT, standard now in the industry. What I'm really proud of are our toy models. So we have a lot of different models of all of our reaction vessels on scales, so with different geometries, and the agitators gives us a good idea about the geometrical situation we have on scale, and makes the upscaling a bit more successful. This is based on a large list on analytical technologies we have available. So the list is quite long, but you know that's solid state chemistry. You need a lot of analytical tools. So enough about CARBOGEN. Let's directly start with today's talk from isothermal solubility data. What is the purpose of translating this data? As you all know, Crystal 16 data, or just the classical solubility profiles, look like this. So you have a certain concentration, which is a solubility as a function of temperature, and you can plot this in an exponential fashion. It will give you an idea about how systems behaves, how systems supersaturate, but it's always for a fixed composition. You all know that a change in composition can change solubility profile dramatically. This is a real case where we had something in IPA, quite a nice deep solubility profile, good for cooling crystallization. But as soon as we started adding TBME, it got very flat. Polythermal solubility data is quite useful. For example, what we have here, it gives you an idea about the degree of saturation you have in your system. So, for example, This is real data from our lab. So here at approximately 80 degrees Celsius and 200 milligrams per milliliter, if I only cool down 10 Kelvin, I get a massive drop in solubility, delivering me approximately 14 milligrams per milliliter in Kelvin of supersaturation. If I do the same thing at 60 degrees Celsius to 40 or to 50, something like that, I only get back, yeah, that is more than 10 Kelvin. My apologies. But as you can see it graphically, it's dramatically less. However, but what happens if my system composition changes? So during anti-solvent mediated crystallization, I have this curve changing all the time. What can I do about it? We can start with a little bit about methodology. First of all, if I'm doing an anti-solvent mediated crystallization, I need to define the solvent and an anti-solvent, so I need to get a rough idea about my solubility, for example, from existing data, or I just do a little screen with a HPLC. But it's mandatory to have an idea what solvent and anti-solvent you want to use. Next step is to set up a 4x4 composition matrix. This is a wild tray, so you get it delivered with the crystal 16. And for example, you can say, okay, I have always the same composition and solvents in row A, 20% anti-solvent, then 30, 40, 50 for rows for different compositions. And down here with the numbers, always the same concentration. So 50, 100, that's always 50 milligrams per milliliter, 100, 200, 300. For example, something like this. And you will end up with a matrix which could look like this. So you'll have the same composition in anti-solvent here and the same concentration down here. How to do this, or how accurate does it have to be? Because it should be always approximately the same concentration. It doesn't need to be too accurate. I show you why afterwards, but please don't waste too much time and make yourself angry sitting in front of the balance a whole day. It just, for my rule of thumb, it's okay if you're weighing in more than 100 milligrams, go plus minus 3% or 3 milligrams, and if you're lower, 2 milligrams plus minus, it's absolutely enough. For me, my target is to have a whole set, so 16 concentrations prepared in maximum 2 hours. And if you're lucky as I am, maybe you have an automated powder dosing system, like a Quantus machine from Mettler. So for some substances, when it works, it can really save you time. Yeah, quite cool thing to have. So data acquisition, you have now prepared 16 samples, and you will put it in your Crystal 16 device. And with that, sometimes it comes with a couple of pitfalls. So you have to check your data quality before you start processing it. For example, you can have decomposition. So this in orange, we have the transmissivity data, and in blue, the temperature data. So as your cell is warming up, at some point, you will have a clear point, so your material goes into solution, and at some point it will reprecipitate, typically at lower temperature when it goes into solution. If you warm up again and you see, okay, it's 2 degrees lower, you might face already decomposition, and in a third cycle, it's even lower. Okay, you'll have a problem with decomposition. You should check with HPLC or potentially alter your heating profile. You also see then the material stays longer in solution or doesn't even precipitate in the measurement anymore. Next one, which I already had before, not that often, but it can happen, is polymorphic form conversion. So I'm warming up, same situation as before, clear point of 40 degrees Celsius, but the next one is a 45, and the next one again is also 45. So I have different solubility here. than I do have here. So this is an indicator for the formation of thermodynamically favored form compared to what I had here. I really have to question this data. Maybe there's also form conversion ongoing down here. Can happen, not too often. Agglomeration is ugly, but sometimes we have that as well. Can come with melting or material properties. So sometimes you see a very long dissolution phase. Sometimes it's even only dissolved when cooling down. We saw this a couple of times where the material seems to be only dissolved when cooling down, just because the agglomerates made the dissolution so slow. General approach for me is to start at 20 degrees Celsius if possible. If I have to start at lower temperature, that's possible with a crystal 16. I do not recommend to use cold solvent. I rather recommend to start the crystal 16 program at minus 20 degrees Celsius with no agitation, the empty machine. In parallel, I add my solvents very carefully on top of my solid. I crimp my vials, and then I put it very carefully in the pre-cooled machine and let the vials cool down for 1 hour. This will allow the vials to get as minus 20 degrees. Then I start agitation in the heated. Works quite nicely to have good data for very low temperatures. And then my rule of thumb going up with 0.1 Kelvin per minute for heating and cooling much faster gives you fines which have better dissolution properties. But also can trigger other polymorphic forms, but that's a whole different story. When your data looks like this, and this is something I just recorded 2 weeks ago, you're happy, so sharp transmissivity signal goes up and goes sharp down again. So this is really how it should look like. I have now acquired all my data and I can plot it. This example here is in DMF water. It's a real life sample. I will talk a little bit about this more today. It's a 3 times 4 sample matrix. It took me approximately 1 hour. And I can see here nice little curves. It's only 3 points. I'm absolutely aware of that, but it's very low data. The data amount is low. What I can see here very nicely, okay, 20% water in DMF is the best solubility, increasing 30, 40, 50% of water in DMF. So the increasing amount of water is giving me an increasing, a decreasing solubility. For this example, I see that water doesn't flatten my curves. It could be that the last one looks like this and is completely flat. It is not. I think this is really good for an anti-solvent crystallization because it allows me to control via cooling in the end. And water reduces my solubility, so this is exactly what I expect. So I wouldn't say that I have any hydrate formation or something like that. These points can be approached via exponential function. So typically the best way to approach is the 1/2, but you can also put in just a classical exponential function engine. Now what I do to transfer polythermal into isothermal solubility data is that I use a semi-log plotting of the measured concentration. So semi-log plotting of the natural logarithm of my concentration versus the reciprocal temperature. This is exactly as what we would do for an Arrhenius plot in physical chemistry for kinetic data. If I do this, I get linear graphs, a couple of them, for my composition. So 20%, 30%, 40%, 50%. These linear graphs will also have some regression parameters. And now it starts getting interesting because right here, I marked the different temperatures. So 30 degrees Celsius will be here. And this will allow me to extrapolate data because I could just extrapolate this set of data into 30 degrees Celsius. I know that it's dangerous, but we can do it. And I show afterwards what the benefit of that is. But I have now the option to set isotherms here. So this is temperature, and see where these axes are crossed. So I have now for this isotherm, I have a data point of 40% water and 50% water. 40 and 50% water, that's real data. And this gives me then a concentration. So it's an ln here, I know, but we can just turn it into a concentration quite easy. I have some text here for the slides will be shared afterwards if someone wants a little bit to read again. It's possible to extrapolate, and I will do it in this example, but it's better to have more data for sure. But this example was quite quick and dirty, and it did the job. If I go back now, and I have now my isotherms, so this marks an isotherm, and I have two real points here, two real points here, two here, and one here. And I plotted and I marked here the real data points, and I extended with the extrapolated data more than 50% in this case. I get this picture. So now I have isotherms. So this is my isothermal solubility data. So I have my solubility as a function of amount of water here in these isotherms. The 70-degree isotherm is a big care, should be handled with care, because high temperature typically makes what it wants. And always, please be careful, only use such models as for an estimation of an initial experiment. It doesn't replace proper experimentation, but it can really give you an idea on what's going on in your system. Okay, based on these data, I can now start thinking of a process design. I showed you solubility data for a compound which comes out of coupling reaction, classical peptide chemistry with TB2 and DMF. Also a little bit of base is present, so this is a historic and validated process at ours, and it works with an extractive workup using DCM and water for removal of the byproducts and DMF. You see here, okay, we start with the TB2, it delivers HOBT, this urea derivative, and this dipea salt. But for a customer, DCM starts to get a problem in the US, so we thought of what we could do to remove the DCM, and I said, okay, why not try direct precipitation after the reaction from TB2 and DMF? And a direct precipitation was initially tested, but was really hard to get under control. But then I measured this very fast, gathered solubility data on the Crystal 16 device, presented to our customer, and the customer agreed to reactivate this investigation. So this is a little bit of another data set. It's so the data set I showed before was with a lot of extrapolation was from the preceding step of that one, but I would now talk a little bit about that one because here we have a little bit more data to discuss. I did the same thing as before, a little bit of less extrapolation of data, but still we have the isotherms here in DMF water mixtures. And here my products, I have 25 grams of product approximately in 130 mils of reaction mixture. Gives me roughly 200 milligrams per milliliter. There's some DIPA in it and some salts. So it's this data is on acquired on the same pure product. So I would expect that the real system behaves a bit different. But what I do now is thinking of a dilution line. So my hypothetical situation is I start here adding the anti-solvent, and my maximum concentration in the system is going down. As I'm adding water, the volume increases, but my 25 grams of product still stay the same. And if I go to a 50% amount of water, I will exactly have the maximum concentration in my system. And when crossing this line, I know that I'm getting into the region of supersaturation. So if I'm doing this addition of water, I can add my water here. I cross my 6-degree isotherm at approximately 24% water, 23% water. I will add a little bit more water. Goes down here quite steep, so I want to be careful. Then I add seeds, potentially age, and then continue dosing water very carefully, high temperature, and then cooling down and harvesting. I really like to do anti-solvent mediated crystallizations at high temperature. So mixing is better, growth kinetics are better, and you typically have less inclusions. So it's always, in my eyes, always better to do this at the highest possible temperature. Sure, you have to consider decomposition. How did the first shot look like? We saw an excellent desaturation here. So in pink, we have the crude product. In blue, the mother liquor with all the very polar material here. In addition to DMF, and purging was very well into the mother liquor, so we took care. However, as you can imagine, water and DMF system, it showed us challenging filtration and drying properties. We suspected a level high supersaturation. This material came out quite quickly, so there must be an influence of salt and base, not yet completely understood. We also see agglomeration, which prevents needles from growth. So we have a high fine count, slow filtration in the lab, and some amorphous fractions were spotted in the PLM. Next steps for us is to investigate the seeding point. We'll go back with the amount of water added, a higher process temperature, potentially checking the rate of anti-solvent addition, and also we will investigate whether thermocycling is an option or not. The second real life situation is about recrystallization. It was quite high volume from ethanol and water, 35 volts. So that was just made to keep the material in solution for an inline filtration. So the material was dissolved at 80 degrees Celsius, almost refluxed and inline filtered. Then followed by distillation, which ended up with the precipitation of the material. It's an old process. It was never touched, but it was not validated because it's not in a GMP sequence, but it poses an unnecessary blockage of vessel capacity. So we were asked to intensify this process. I did the same thing as before, recorded polythermal solubility data. We had a couple of data sets before. That is the reason why this is, I was very lucky. That was my first shot. So from 16 data points, I got out 15 clear points, just 150% water in ethanol. Wasn't recorded. However, we see quite interesting behavior here. So the best solubility here is 20% water, followed at 35% water, followed by 5% water, followed by 50% water. If I translate this now in isothermal solubility data, I see a synergistic solubility. So I have the highest solubility for this compound at 25% water. That's quite interesting. That's not something, that's not wrong data because it's based on 16 single data points. So I know this is reality. All single data points show me that approximately in this region must be the synergistic solubility peak. Quite interesting, quite good to know. Based on that, we said, okay, we build a process. We started 25% water. That's only 60 degrees Celsius here. So we go approximately to reflux to dissolve it. We do an inline filtration hot, 70 to 75 degrees Celsius, and we saw from the nucleation data that we have an operation window of 20 to 30 degrees Celsius. The warmed inline filtration was in temperature 75 degrees Celsius, and we added water to achieve a water content of 58 degrees Celsius, 58%. And then we define the cooling, the seeding spot. And here I showed that from a first trial because I don't have the data for the 58 volume percent water in ethanol. But if you would have stick to the 50% process, which was what we initially planned, we can directly go back in our polythermal data. So the polythermal data is still quite useful. So if you would decide here to only go to 50%, what we did initially, but wasn't very successful in terms of yield, we could use this data to calculate the seeding point. So we calculated here supersaturation of 25%. That's quite accurate for recrystallization in meat systems. But we had to move away from that because the yield was a bit low with 85%. Slurry was a bit not enough mobile. So the final process was then containing higher levels of water. At the end, I would like to speak a little bit about the limitations. As you saw, it's not very accurate. It's only very good for idealized systems, but it can give you a good idea. You should have some solubility data prepared. Otherwise, it can be quite laborious. But maybe it's also good to have a short screen before you start with work like that. And it doesn't replace proper lab work. So based on the data you have, you should make the right decisions for the choice of your parameters. It gives you no information about your growth behavior or your filtration drying behavior. And upscaling still needs to be handled with special care. What is the benefits? As said before, it can be a door opener to motivate management or your customers for process improvements, even if they don't want to. It has a very little turnover time, so 1 or 2 hours preparation, and you have the data available next day. You don't need too much material, and it's a fast method to accrue the process. So for the recrystallization, I showed we only had 2 development experiments, and now the upscale run is pending, but I don't really expect any surprises. And it can give you insight about whether your anti-solvent reduces your solubility fully proportional or not. If you have synergistic solubility, that's definitely something you want to know. Then that was already it from my end. Thank you very much for your attendance.
Thank you very much, Marco. Really nice presentation. Amazing to see how you start from a very small scale, the crystal 16 or even lower, and then you go towards the liter scale. I had a question. I noticed. Oh, somebody raised the hand, but I'm not very sure if I can allow anybody to talk live. So please write to us the questions in the chat function. Then it's much easier. No, I saw quite some troubles or challenges, let's say, around aggregation. I do believe you have quite some experience with it. Are there any ways on how to go around it or to minimize it? I'm asking this because just in the morning we had a question from a customer from Japan related to agglomeration, and yeah, I also have a bit of experience with it. Yeah, it's also some compound related sometimes.
Yes. So, oh, sorry. Yeah, my presentation makes what it wants. You were asking about this image, right? So if you have agglomeration during your measurement, there's not that much you can do.
Yeah.
What we learned, so There's a model substance I can talk about it. That's glycine. We know polymorphic form from glycine, the gamma form, in the presence of water and sodium chloride, tends to do large agglomerates. And the problem here is that the agglomerates turn so big that the laser, or the LED doesn't really think that it is a slurry anymore. So it's just getting big chunks. And uh, so what we do, and that's if we see something like that, we suspect agglomeration. We stand next to the machine. We take the vial out, and we see the snowballs flying around. You can try to flatten your heating a little bit, or what we sometimes do is we do it in a reactor with IR solubility measurement, or even even better, we have a camera which makes a movie next to the reactors, so we can check with our bare eye what is going on. Sometimes technology is it's you see ghost signals. In the
Yeah.
crystal 16, if you have strong agglomeration, I don't have any
No,
remaining
indeed.
solution.
Yeah, you will see definitely in the signal, in the transmissivity,
Mhm.
but yeah, there's not that
Yeah,
much.
yeah.
Yeah.
You sometimes you see transmissivity signals going a little bit up, a little bit further, and you don't
Yes.
really know. Okay, do I really have to take the 100% signal, or what is going on? It's just data I cannot 100% trust.
Yes. We do have somebody is asking, what is your source for getting ideas for promising anti-solvents? So how do you decide on the anti-solvents, the good ones?
I start typically with HPLC data. So I'm making a solubility screen at 20 degrees Celsius. So I make slurries, 20 slurries, then overnight I let it shake overnight in a shaker, centrifugation, take the take the supernatant, make a quick HPLC with a calibrated HPLC method, check the solubility, do the same thing at 40.
Yeah.
And yeah, there's sometimes you see always the same candidates, like very unpolar ethers or heptane, something like that. I try it. Anti-solvents, I think, um, toxicity is really important when it comes to late stage. So water is always good if you have alcohol and water systems. Heptane can be good, but it has to be handled with caution. Toluene can be good, but has to be controlled. I like low boilers, but yeah, that's
Yeah.
it's often a gut feeling. Sometimes what we also do when we make a centrifugation, and we see, okay, we have a couple of solvents which show low solubility. We check in a microscope how the particles look like, whether they changed a
Yeah,
little bit
that's a good
their
one.
morphology, and we can get good morphology of morphological information. Yeah, so stuff like that. It's from different sources.
Yeah. Slide 22, Marco. Is there a good reason to not stabilize the temperature at 60 degrees during heating cooling ovens?
No, I just make this in Excel. I typically stabilize the temperature. Absolutely right. I just wanted to have this
Nice.
two, yeah, in Excel to move around the temperatures to show the effects. But that was handmade in Excel. Yeah, absolutely right.
When you have the Arrhenius type plots, do you
Yeah,
convert to molar units or in weight percentage?
no, um, that has the reason. So if you go to molar units and you change your composition, it is very tricky. So I did it a couple of times and checked, but I stay with a concentration. So yeah, if I have something like that, no, I don't change to mole per mole. because yeah, it's getting complicated if you have changing composition. I hope you understand what I mean.
Yeah. Have you thought about plotting temperature, solubility, anti-solvent into a 3D survey?
Yes, yes.
And then develop a crystallization road from there?
Yes, no, that's possible. You just need to have the right software. I think
Yeah.
for a presentation, it makes most sense to have it like this, or yeah, for example, you can plot this as a surface, or you can plot this as a surface. It's possible, but if you have a surface, it's cool if you can move it around, you know.
Yeah. Indeed. Yeah.
I made contour plots. It's also okay, but yeah, it's there's software available capable of that. You can use your classical DOE software, just use it a little bit otherwise and change the factors into parameters to your solubility data. Will give you nice contour plots. Excel is not that easy, but the DOE software typically can do that.
In the first example, has your team tried other solvents apart from DMF? Sometimes acetonitrile can be as good as DMF, and it's a bit less costly.
Yes, would be maybe in the past we could have tested that. The thing is that there were tests for the reaction, and DMF was considered the best and was not changed. And the thing is, this process is validated. So what we can do is cut away the workup section and say, okay, it is a validated process, so we leave away the workup, but if you want to change the reaction conditions, for example, the solvent, the whole validation has to be repeated. And this barrier is too high. But I know there was other solvents tested, but that's not something I can take an influence on now.
How many percentages of the desired polymorphic form achieved of the product expected with adopting this technology? Yeah.
I don't know. It's I can't make any statistical
Yes.
statements. It's difficult to say. So 100% in my cases, but yeah, the control of the polymorphic form is often only important on last stage for an API if it is an oral application. So for intermediates, we typically don't control the polymorphic form until it doesn't dissolve again. And the next step, then sometimes we open this box. But I'm not sure.
Somebody's asking, with anti-solvent type of crystallization, can we use a syringe and use an anti-solvent type of reactor cap for generating the solubility as function of anti-solvent directly? I think for the crystalline, yes. For the crystal 16, you do not have a cap for the anti-solvent.
That's you know that better than I do. So I have fixed composition in my vial.
Yes, yeah. And with the crystalline, there are quite a few methods developed on how to look into anti-solvent type of crystallizations. If interested, look on our Ankit. Look on our website under crystallizationsystems.com under publications, select crystalline, and you'll see there the white paper on solubility. And there for sure we'll have a chapter on detailing that. Now, going back to this, I'm reading to you, Marco. Thank you, Marco, for the great presentation. Have you experienced fall information in the crystal 16 vials? Like a film, I guess, on the glass, which prevents proper mixing. Indeed, this is, I don't know if you have experienced it, Marco, but it's also compound dependent. It's also we used to use also silanized type of vials to overcome some of this. And definitely the third version of the Crystal 16, the latest one, the reactors are designed in such a way that you have also a colder upper part that will prevent this. The same is also the crystalline version 2 design. Will
So if I have experience on detecting films and coatings, yeah, not really. So if we see that we need to put in more energy, we use the steel hooks. But often for this kind of investigation, the solubility data is more important for me than nucleation data. And I would assume that film or coating is something linked with cooling down or nucleation. If I fill in a powder and warm up and I have formation of a film, then my problem is a whole different one.
Yes.
If my material melts in the measurement or something like that. That's something I saw in melting of a substance, because it seemed to be completely decoupled from whatever we do. It dissolved always 60 degrees Celsius, and then we recognize that it's melting. But during this melting process, so things can happen. But often, when I see something happen weird, block formation or coating, that's linked with the nucleation part.
Yes. Have you ever experienced polymorphic conversion and you were able to detect it by transmissivity profile change? Like
yes.
Hydration, solvation? Yes.
So there is this, the one I showed before, when, okay, what can happen is that your clear point is going up from measurement to measurement. But what we saw before in one project was a quite weird behavior. I don't have it in the presentation right now, but we saw that the transmissivity data was going up and then stalling and then going up again. So it really was going up, wanted to dissolve, reprecipitated, and then went up again.
Yes.
So in the transmissivity data itself, we saw such a knee. And that was our indicator for a polymorphic form. And what we did when we saw this data was measuring powder X-ray before and after. And we saw that we had certain reflexes going down. So we started with a mix of a polymorphic form. We didn't care too much about polymorphism of the starting material because it was an intermediate, but our data was quite scattering, and it was really, the data changed a lot as function of the heating we applied. So when we were heating very slow, we see lower solubility than when we were heating fast, but in a atypical way. So you have to be cautious about the data. If you don't see this, you can ask yourself, okay, what's going on?
Yes, indeed. This is also a bit the beauty of the Crystal 16, that this maybe is just a line going up and down, the transmissivity, I mean, but in many cases can tell you quite a lot about what is happening. But of course, you always need secondary analyses and to go more deep and understand exactly what is going on. But definitely the first indications you could have. I have one more question and a lot of thank you. Do you always perform 3 temperature cycles for your Crystal 16 data acquisition? Or what is no? Okay.
No. It really depends. If it has to be really quick and dirty, I do just one. I warm up, cool it down, and then take the data as is. If it has to be like for a little bit if I present the work to a customer, I take 3 cycles as default, yes, and sometimes I take more for certain reasons. If I really want to be sure, if my solubility data has to be very bulletproof, I take it with different heating rates and cool down quite fast and go up with a different heating rate and do this different alterations just to have bulletproof data if I want to make a major process change based on this data. But sometimes just quick and dirty. It's
yes,
so fast.
indeed. It depends on what is your objective, what you want to get out of it. Yes. One last one. I said already the last one, but I see one more. So from Pedro. Can you reuse this data to create population balance models for crystallization modeling development?
I wouldn't recommend that. Because for PBM work, I think the quality of your data has to be really good. Because yeah, you know, shit in, shit out, and I'm not really sure. I'm not working too much with PBM, so I'm not sure. But I think the quality of the data I show here, the way of acquisition, is not accurate enough. Maybe
We do have.
maybe yeah.
Sorry, go ahead.
Try it. Maybe it works, maybe not.
No, we do have, we had in the past, I'm not very sure, I think last year, a webinar with Professor Gerard Capellanes from Rowan University, US. And he's quite known. He even developed a piece of software, or he's working on that, on population balances, and he is in this, and he also has at least one publication, an article, scientific publication that I know about it on population balances. In one of his articles, what he's using there for population balances is data from our crystalline instruments, so data taken out of the images that you can collect with a crystalline. So if you're interested, you can again look in our database of webinars, and you'll find it there. The webinar on population balances from Gerard Capellanes. I think this is it, Marco. We have covered quite a lot, and as I mentioned to you, I already see quite a few. Thanks, Marco. Thank you. Great presentation. So I think everybody was happy to join. I am happy that you were here and you brought this topic to our attendees. I think you presented a year ago.
Yes, yes.
And I'm really thankful that you agreed also to do it online and to reach more people. You see the hands. Thank you very much. If any questions, anybody still after this webinar, please feel free to contact either Mark or either ask at info@crystallizationsystems.com, and we will do our best to answer or to share your questions with Marco and to answer you. We will be back, Technoviz will be back with another webinar soon. And this is it for now. If you are in Europe, have a good afternoon, evening. And from our US attendees, have a good day. Thank you very much, Marco, once again. Bye.
Thank you. Goodbye, guys. Bye-bye.